Master Genetics for NEET. Decode NCERT with us. Score 360 in biology. Your dream medical college awaits. Subscribe now and let's crack it.
When we trace the origin of plants, we are tracing how microscopic single-celled organisms in the water eventually engineered the most massive structures on Earth. Early taxonomists, like Carl Linnaeus, grouped organisms using surface-level characteristics. If a biological specimen had a rigid cell wall, it was classified as a plant. This system meant organisms like fungi and cyanobacteria were lumped into the same category as oak trees. Modern biology uses phylogenetic classification. By mapping deep internal structures, embryology, and evolutionary ancestry, we redrew the boundaries. Cyanobacteria and fungi were excluded from the plant kingdom entirely. The modern taxonomic divisions act as a sequential record of the exact structural engineering required for aquatic organisms to conquer dry land. Moving inland presented two massive hurdles. The first is gravity. Without water to provide buoyancy, plants required structural plumbing to hold themselves up. The second is desiccation. They needed a way to reproduce without their sex cells instantly drying out in the open air. To see how they solved this, we have to look at the baseline of plant reproduction. Plants alternate between two distinct phases: a haploid gametophyte, which has one set of chromosomes and produces gametes, and a diploid sporophyte, which has two chromosome sets and produces spores. The evolutionary journey from aquatic algae to towering trees follows an inversion of which of these two phases dominates the organism. Solving the engineering problem of dry land required plants to restructure their internal anatomy and reconfigure their entire life cycle. We start with algae, the aquatic baseline. Algae lack true roots, stems, or leaves because they simply do not need them. The surrounding water provides immediate physical support and allows nutrients to diffuse directly into their tissues. Because they lack complex terrestrial tissues, algae are classified by their chemical engineering, specifically their photosynthetic pigments and how they store food. This creates three distinct classes: the green Chlorophyceae, brown Phaeophyceae, and red Rhodophyceae. These chemical variations, like possessing chlorophyll A and C versus A and D, allow different classes to optimize light absorption at different oceanic depths. We see this manifested in the distinct physical forms of the green Volvox, the brown Fucus, and the red Porphyra. Algae mastered aquatic photosynthesis, but their lack of structural tissue and strict reliance on swimming spores keeps them permanently tethered to the water. Moving to the shoreline, we find the bryophytes, the mosses and liverworts. These plants can survive on damp soil, acting as the amphibians of the plant kingdom, but they remain strictly bound to humid environments. Their vulnerability lies in a single biological restriction. Their male gametes require a physical film of liquid water to swim to the female sex organ. In bryophytes, the free-living green plant is the haploid gametophyte. The diploid sporophyte is a secondary stalk growing directly out of it, siphoning nutrients. Its only function is to cast haploid spores to generate new gametophytes. Bryophytes successfully breached the land, but without internal plumbing or dry reproduction, they are trapped as diminutive shoreline dwellers. Breaking the height limit required new technology. Pteridophytes, like ferns and horsetails, are the first terrestrial plants possessing dedicated vascular tissues, xylem and phloem. These internal pipes allowed plants to pull water up from the soil and push nutrients down against gravity. This physical transport system enabled the evolution of true roots, stems, and complex leaves. This structural leap required a life cycle inversion. In nature, the massive fern is the diploid sporophyte. The gametophyte has been reduced to a microscopic, flat prothallus. But pteridophytes face a massive evolutionary paradox. Despite their towering vascular height, they still rely on that tiny, fragile prothallus in the damp soil to facilitate swimming gametes. They solved the structural engineering problem of gravity, but their water-dependent reproduction still limits their geographical spread. Conquering dry terrestrial life required a plant to sever its reproductive reliance on liquid water completely. Gymnosperms, the naked-seeded plants, introduced the ultimate reproductive innovation. They took that vulnerable male gametophyte and shrank it down into a highly portable, microscopic package, the pollen grain. Instead of living independently on the ground, the female gametophyte is retained entirely within a megasporangium, the ovule, protected directly on the exposed cones of the tree. When an airborne pollen grain lands, it physically grows a pollen tube directly into the ovule. This tube delivers the gametes straight to the egg, completely replacing the need for swimming sperm. Angiosperms act as the final iteration, enclosing their ovules inside specialized ovaries, flowers, and fruits to manipulate animals for targeted dispersal. By miniaturizing their vulnerable gametophytes into pollen and ovules, seed plants eliminated the need for surface water entirely, allowing them to inhabit the entire globe. The taxonomy of the plant kingdom provides a direct ledger of terrestrial engineering milestones. This chart shows the reproductive shift across all 4 taxonomic divisions. In algae and mosses, the haploid gametophyte dominates the mass. As we move toward the vascular ferns and seed-bearing trees, the diploid sporophyte exponentially grows until it overtakes the screen. To grow larger and conquer the planet's surface, plants had to shrink their sexual generation until it became microscopic. True planetary dominance relied on building upward with vascular plumbing and fiercely protecting the fragile origins of the life cycle.
Great job on completing this lesson. Click here to keep learning more. Check out these recommended videos. Let's crack your NEET exam. See you next time.